replication is the process by which DNA makes a copy of itself during cell division. The first step in DNA replication is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks the hydrogen bonds holding the complementary bases of DNA together. The separation of two single strands of DNA creates a Y shape called replication fork. The two separate strands will act as templates for making the new strands of DNA. One of the strands is oriented in the 3 to 5 direction. This is the leading strand. The other strand is oriented in the 5 to 3 direction. This is the lagging strand. As a result of their different orientations, the two strands are replicated differently. Once all of the bases are matched up, an enzyme called exonuclease strips away the primer. The gaps where the primer are then filled by yet complementary nucleotides. The new strand is proofread to make sure there are no mistakes in their new DNA sequence. Finally, an enzyme called DNA ligase seals up the sequence of DNA into two continuous double strands. The result of DNA replication is two DNA molecules consisting of one new and one old change in nucleotides. This is why DNA replication is described as semi-conservative. Half of the chain is part of the original DNA molecule, half is brand new. Following replication, the new DNA automatically winds up into a double helix.
So the next process is transcription. The process of transcription begins when an enzyme called RNA polymerase attaches to the template DNA strand and begins to catalyze production of complementary RNA. Polymerases are large enzymes composed of approximately a dozen subunits, and when active on DNA, they are also typically complex with other factors. In many cases, these factors signal which gene is to be transcribed. There are three types of process in transcription. So the first one is initiation. So this is the first step in transcription. When the RNA pol binds to the DNA upstream of the gene at a specialized sequence called the promoter. And the second one will be strand elongation. Once transcription is initiated, the DNA double helix unwinds and RNA polymerase reads the template strand, adding nucleotides to the three end of the growing chain. At a temperature of 37 degrees Celsius, new nucleotides are added at an estimated rate of about 42 to 54 nucleotides per second in bacteria, while eukaryotes proceed at a much slower pace of approximately 22 to 25 nucleotides per second. So the third process is termination. So terminator is the sequence are found close at the end of non-coding sequences. Bacteria possess two types of these sequences. In row-independent terminators, inverted repeat sequences are transcribed. They can then fold back on themselves in hairpin loops, causing RNA pol to pause and resulting in release of the transcript. On the other hand, row-dependent terminators make use of a factor called row, which actively unwinds the DNA RNA hybrid. So, in eukaryotes, termination of transcription occurs by different processes, depending upon the exact polymerase utilized. For Pol1 genes, transcription is stopped using a termination factor. Through a mechanism similar to row-dependent termination in bacteria, transcription of Pol3 genes ends after transcribing a termination sequence that includes a polyhierarchical stretch by a mechanism resembling row-independent prokaryotic termination. Termination of Pol2 transcripts, however, is more complex.
Translation is the second part of protein biosynthesis, or the making of proteins. It is part of the process of gene expression. In eukaryotes, translation happens on the ribosomes in the cytoplasm and in the endoplasmic reticulum. In bacteria, translation happens in the cell cytoplasm, wherein they have no nucleus. Translation is the process in which ribosomes in the cytoplasm or synthesize proteins after the process of transcription of DNA to RNA in the cell's nucleus. In translation, messenger RNA or mRNA is decoded in the ribosome decoding center to produce a specific amino acid chain or polypeptide. The polypeptide later folds into an active protein and performs its function in the cell. The ribosome facilitates decoding by inducing the binding of complementary tRNA anticodon sequences to mRNA codons. The tRNAs carry specific amino acids that are chained together into a polypeptide as the mRNA passes through and is read by the ribosome. Translation has three phases. The first phase is initiation. The ribosome assembles around the target mRNA. The first tRNA is attached at the start codon. The second phase is elongation, wherein the tRNA transfers an amino acid to the tRNA corresponding to the next codon. The ribosome moves or translocates to the next mRNA codon to continue the process, creating an amino acid chain. The last phase is termination, or to stop. When a peptidyl tRNA encounters a stop codon, then the ribosome folds the polypeptide into its final structure. The three phases of translation initiation, polymerase binds to the DNA strand and moves along until small ribosomal subunit binds to the DNA. Elongation is initiated when the large subunit attaches and termination ends the process of elongation.